Double bond comes in between the carbon atom or double bonds are repeated in between the carbon atoms, then it is an unsaturated fatty acid. So those 2 points you can write. And the alpha carbon atom also has a variable group, right? What is that variable group? It is the R group. And on the side chain they also have a hydrogen. Two monosaccharides are attached to each other by glycosidic bond. Amino acids are linked to carboxyl group, its amino group and all that through peptide bond. Hello everyone, a warm welcome to this session on revision class for first PUC Biology. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session of revision class, we learn about biomolecules. That is, in the previous session, under this particular chapter, we learnt about the synopsis, that is what exactly are the contents that is present in that particular chapter, and also we learned some of the important multiple choice questions that are likely to appear from that particular chapter. So in this session, that is session 2, which is a continuation of the previous session, we will learn 2 marks question and answer. That is, 1 2-mark question and answer will be asked from this particular chapter and 1 3-mark question and answer will be asked from this particular chapter. So both of these 2 marks and 3 marks, let's discuss. So 2 marks question and answer. So name the inorganic compounds found in living organisms. So there are 4 inorganic compounds which you have to mention here. So 4 if you write, each will carry half mark, therefore you will get 2 marks. So it is water, salts, acids and then bases. Next question, write the difference between fats and oils. So this is one of the very important question. So difference between fats and oils. So whenever I told you, whenever the word difference, differentiate, all that comes, you have to make a column. On one end you need to write fats and on other end you need to write oils. And whenever you write the differences, it should be just the opposite of what difference you're writing here. Say for example, here the first point that I've given under fats is they remain solid at room temperature. So here also you have to write the opposite of it. Here what it is the difference? They remain liquid at room temperature. Next, fats, they are saturated and trans. 2 types are there, saturated and trans fats are present. In this, unsaturated fats like monounsaturated and polyunsaturated are the types of unsaturated, that is fats. So oils are nothing but unsaturated fats. Fats are nothing but saturated. Next, fats, they are mostly derived from animals, whereas oils are mostly derived from plants. This point is quite easy because all the refined oils, if you remember, the coconut oil or be it the cooking oil that you use, all that are derived from the oils, whereas fats are usually derived from animal source. So any 2 difference if you write out of the 3 is also enough. So moving further, these structures, please study the structures from this chapter, whatever structures are given, be it for amino acids, be it for the structure of fats, be it the structure of nucleotides, nucleosides and all that, because usually question can appear. So write the structural formula for amino acid alanine. So we know that the simple structure for amino acid is it contains an alpha carbon atom. On one end it contains the amino group. On the other end, it contains the carboxyl group, and on the side chains, it contains the hydrogen, and on one end, it contains the R. Now here, R is nothing but the variable group. Only the variable group will change here. So if it is alanine, it will be CH3. So therefore, if you learn the structure, that is simplest structure of amino acid, now rest of the things you can write. So alanine means here it will have an alpha carbon atom. Same, on one end it will have the amino group, on the other hand it will have the carboxyl group, one more it will have the hydrogen, and then on the R group, that is the variable group, will be methane here, that is CH3. Then it is called as an alanine. So much if you write, you will get 2 marks if you write the structure. Next, what is the structural formula for amino acid? Serine. Serine means it should be CH2OH. So the variable group, if you write it as CH2OH, it will be serine. Next question. Write the structural formula for amino acid glycine. They'll ask only these three structural formulas of amino acid because only these three are given in the NCERT textbook. That's why. Beyond that, they will not ask. So what is there in the NCERT textbook? Only that if you study it is enough. So for here, you have to add a hydrogen. If H is added, then it will be a glycine. The variable group will be a hydrogen here. So next question, write any two differences between saturated and unsaturated fatty acids. So saturated fatty acids and unsaturated is the difference that we have to write. So saturated fatty acids, they do not have a double bond between the individual carbon atom. So fats are made up of chains of carbon atoms linked either by a single bond or double bond in between. Now, if they do not have a double bond in between the carbon atoms, say for like this, if it is like this without any double bond, then it is a saturated fatty acid. Here what happens, at least one double bond will be present. Somewhere at least one double bond will be present here. So if double bond comes in between the carbon atom or if double bonds are repeated in between the carbon atoms, then it is an unsaturated fatty acid. So those 2 points you can write. So here they lack a double bond. Here at least one double bond will be present between the carbon atoms. And saturated fatty acids, they tend to remain solid at room temperature and they are from animal sources. Unsaturated fatty acids, usually they remain in liquid form. That is your refined oil and all that, cooking oil. So cooking oil and all that, during winter, do they get solidified? No, they do not, right? But fats usually get solidified. So therefore here, unsaturated fatty acids, they usually remain liquid during even at the room temperature or at colder temperatures also. So that's the difference. So in two differences each you have to write to fetch you 2 marks. So next question, what are phospholipids? Give an example. This is also one of the very important question regarding phospholipid. Give an example. So what are phospholipids? The name itself says phospholipids. So what? Lipid molecules which are attached to a phosphate group are called as phospholipids. So it is there in the question itself, the answer is there. Now example is lecithin. So lecithin is the only example that is given in your NCERT textbook. So one example you can write here, that is lecithin. So if you write what are phospholipids, you will get 1 mark. If you write the example, you will get 1 mark, totally 2 marks. Next, what are nucleosides? Give an example. This is also a very important question. Nucleosides, nucleotides, the structure of proteins, all these are very important questions from this particular chapter. So what are nucleosides? Give an example. A nucleoside is a compound formed by the union of a nitrogen base with the pentose sugar. Say for example, we have a nitrogen base. So what are the nitrogenous bases in the DNA? It is adenine, guanine, cytosine and thymine, right? If it is a RNA, it is adenine, guanine, cytosine and uracil, because RNA will not have a thymine. Instead of thymine, they'll have a uracil. Now, that particular nitrogen base, if it is attached to a pentose sugar, be it a ribose sugar or be it a deoxyribose sugar, then we call it as a nucleoside. Now, example for that is adenosine and uridine. Why I have taken adenosine and uridine is because that is the one that is given in the NCERT textbook. So, that is why. So, adenosine and uridine, any one they have asked. So, any one you can write. Here 2 I have written, any one out of these you can write. So next question, write any 4 functions of protein. So what are the functions of protein you need to write? So the main function is they transport nutrients across the cell membrane and they help in proteins also. Some of the antibodies are also made up of proteins which helps in fighting infectious organisms. And proteins, some of the proteins, enzymes are made up of proteins and they help in performing various biochemical reactions in the cell. And some of the hormones are also made up of proteins itself and they act like chemical messengers wherein they carry information in the form of chemicals from one part of the body to the other. So these 4 functions if you write, each function that you write will carry half a mark, therefore 2 mark you will get. So next question, write difference between nucleoside and nucleotide. This is also a very important question. Almost all that I've put here under this chapter, the ones that I've put here are very, very important. So nucleoside and nucleotide. So we'll make two columns. On one end we'll write nucleoside, on the other we'll write nucleotide. So nucleoside, what is it? It is a compound formed by the union of what? A nitrogen base along with a pentose sugar. So or you can write nucleoside will have a nitrogen base plus a pentose sugar. Here you can write a nucleotide which is formed by the union of nitrogen base, a pentose sugar and a phosphate. So nucleoside means nitrogen base will be there which will be attached to a pentose sugar. Okay. So nucleotide means nitrogen base will be there or nucleoside will be attached. You can write it this way also. Nucleoside will be attached to a phosphate group. Okay, because nucleoside is nothing but what? It is a nitrogen base attached to a pentose sugar. So therefore, what is a nucleotide? You can write a nucleoside attached to a phosphate group. That is also correct. Like that also you can write. And nucleoside is a component of nucleotide, whereas nucleotide is formed by the phosphorylation of nucleoside. Next example under this is adenosine. For this example is adenylic acid. So any one example you can give. So these are the differences between nucleoside and nucleotide. Or separately they can ask, define nucleoside with an example, define nucleotide with an example. Like that also question can be framed. Next question, this is also quite important. Write the cyclic structure of glucose and ribose. So glucose, all these are heterocyclic ring structures is what they have, wherein they'll have 5 carbon atoms and each of them will have a hydroxyl group. So it will have, this will have 6 carbon atoms, C6H12O6. So you have to draw a heterocyclic ring and then you have to mention the 6 carbon, these ends that are there will represent carbon atoms. So 6 carbon atoms and you have to write the number of hydrogen, that is hydroxyl group, the hydrogen. So same thing what is given in the NCERT textbook is what you have to write here. Ribose sugar also. So it is a pentose sugar. It will be C5H10O5. Because it's a pentose meaning it will have 5 carbon atoms. So this is what you have to write. So this is the formula for a ribose sugar, that is C5H10O5. So moving further, what do you call first amino acid and last amino acid in a polypeptide chain? So in a polypeptide chain. So polypeptide chain means that is in the proteins or in the amino acid which is made up of chains of proteins. There what do you call the first amino acid and the last amino acid? So the first amino acid in the polypeptide chain is called as the N-terminal or it is called as the amino terminal, and the last amino acid in the polypeptide chain is called as the C-terminal or the carboxyl terminal. So you write N-terminal and C-terminal, you'll get 2 marks because each of them you have to mention, you'll get 1 mark each. Next question, write any 2 difference between primary and secondary metabolites. This is one of the very, very important questions that usually appears in the exam. So primary metabolites and secondary metabolites. Primary metabolites are essential for the cell growth. That is, they're directly involved in the growth of the plant, whereas secondary metabolites are not directly involved in the growth of the plant, but they are useful for human beings. And primary metabolites are not involved in any defense mechanism in the plant, whereas secondary metabolites are involved in defense reactions in the plant because they'll contain certain toxins and all that which will be released whenever a pathogen or an insect is coming to feed on it. And primary metabolites example are proteins, carbohydrates and lipids. Secondary metabolites, alkaloids, phenolics, sterols, steroids, essential oils, toxins, all these are secondary metabolites. So any 2 differences if you write it is enough. So moving further, write the graphical representation of temperature and pH on enzyme activity. So you have to draw the x-axis and the y-axis and you have to mention the enzyme activity. This is for temperature. So you have to draw the graph same way for that is enzyme activity. During pH, you have to mark pH, then enzyme activity, and you have to draw this graph. So this very rarely they ask, but it can appear as well. So next question, amino acids are called alpha amino acids and substituted amino acids. Justify. Why are amino acids called alpha amino acids? So amino acids are organic acids having an amino group and a carboxyl group. What is that? They have a carboxylic group and they have an amino group which are attached to a carbon atom which is called as the alpha carbon atom, and hence they are also called as alpha amino acids. And the alpha carbon atom also has a variable group, right? What is that variable group? It is the R group. And on the side chain they also have a hydrogen. Because they have a R group which can be substituted with either methane or hydrogen or CH2OH, because it can be substituted and the R group can change, it is also called as substituted amino acids. So that is why amino acids are also called as alpha amino acids and substituted amino acids. So much you write, you will get 2 marks. Next question. Nucleotides. So all these are analysis kind of questions, okay? Because some of the analysis kind of questions can also appear in the exam. It's not that always direct questions will be asked. Here also is one analysis kind of question, like they have given a statement. Nucleotides are phosphorylated nucleosides. Justify. We know that, right? So whenever a nucleotide is attached to a phosphate group, so nucleotide are phosphorylated nucleosides, meaning nucleosides. So what do you mean by nucleosides? It is a nitrogenous base when it gets attached to a pentose sugar, be it a ribose sugar or a deoxyribose sugar, we call it as a nucleoside, right? Now that nucleoside, when it is attached to a phosphate group, we call it as a nucleotide. So that is what you have to put in words. So nucleotides are phosphate esters of nucleosides. The various nucleotides in nucleic acids are linked through phosphate ester groups. That is why nucleotides are nothing but phosphorylated nucleosides. So you can put it like this, or as I explained also, you can write the answer. So you will get 2 marks for that. Next question. Name the 4 types of proteins based on the structure. So based on the structure, there are primary proteins, secondary proteins, tertiary proteins and quaternary proteins. So each one 4 you have to write. So each one will carry half a mark. Therefore 2 marks you will get. So next we will move on to 3 marks question and answer. Any one 3 mark question will come from this particular chapter. So this is, they have given a statement. The statement is, the power of enzyme is incredible. That's true, right? Why do we use enzymes? We use enzymes to catalyze the rate of a reaction or increase the speed of a particular reaction. Say for example, some reactions can produce very little molecules or very little compound, but the same thing, and it may take too much of time also, but the same thing, you add an enzyme to that reaction, what happens? That reaction will get speeded up and the time duration that it will consume will also become very less, and the number of molecules or compounds it will produce also will become more in number. So that is what it means. So the power of the enzyme is incredible. Explain this with synthesis of carbonic acid as an example. In your NCERT textbook, they have explained this with taking carbonic acid as an enzyme example. So here, enzyme accelerates the rate of the reaction, or you can write enzyme speeds up the rate of the reaction, and sometimes what happens, in the absence of an enzyme, the rate of the reaction can come down, or sometimes the rate of the reaction may not apply or may not happen at all. So what do you mean by rate of a reaction? You take 2 substrates, you mix it and you wait for some other compound to develop from that. Now, when you take 2 substrate and you mix it, sometimes if you don't add an enzyme, that compound may not develop at all. So that is what it means. So for example, in the absence of any enzyme, this reaction is very slow, with about 200 molecules of carbonic acid being formed in an hour. So there is an equation that is given. So that is carbon dioxide plus water gives carbonic acid. Now here, if I do not add an enzyme, then the rate of the reaction will be slowed down. So what I can do is, I can add an enzyme, carbonic anhydrase. So this carbonic anhydrase will speed up the rate of the reaction to how much? 200 times more. Maybe somewhere around 6 lakh molecules of carbonic acid I can get. So where is that, like hardly we could have, without the enzyme, that is what I mean to say is, without the enzyme only 200 molecules of carbonic acid can be got. But here what happens, after I add an enzyme, somewhere around 6 lakh molecules of carbonic acid can be produced every second. See how the enzyme has speeded up the rate of the reaction, no? So that is why the power of the enzyme is incredible, is what they say, taking this equation as an example. So that is what I have written here. So this much you write, you will get 3 marks. This is a very important question. Next question, write a note on 3 factors affecting the enzyme activity. So there are various factors that affect the enzyme activity. I have put all the factors here. Any 3 which is easy for you and which you remember during the time of the exam, you can write it down. So one is substrate concentration. That is how much amount of substrate you add will decide the rate of the reaction. Say for example, or will decide the rate of the enzyme activity. So for particular substrate, particular enzymes need to be used. Otherwise the reaction will not occur. So that is what is one of the factors. Next is pH. So each enzyme has its optimum pH. So if the pH is more acidic, or if it becomes basic, or if it becomes neutral, only certain enzymes will perform certain functions. So therefore the pH also affects the activity of the enzyme. Next is temperature. So temperature also, optimum temperature is needed for the functioning of the enzyme. Sometimes what happens, if the temperature gets altered, say for example, if the temperature is too much during the process of reaction, then what happens, the enzymes can get denatured, because at the end of the day enzymes are nothing but proteins and they can easily get denatured at higher temperature. So therefore temperature is also a limiting factor. Next, enzyme cofactor and coenzyme. So sometimes what happens, even though you add an enzyme, the reaction may not occur. At that time you may need to add a coenzyme. So that will also alter the rate of the reaction of that particular enzyme. Next is enzyme inhibitors. So enzyme inhibitors, sometimes what happens, if you add an enzyme inhibitor, it will mimic the substrate and it will go and bind to the active site of the enzyme, therefore preventing the substrate from binding to it and thereby preventing the reaction from happening. So therefore enzyme inhibitors can also affect the enzyme activity. So these are the different factors that are there. Any 3 if you write is enough. Next question. What are the 3 distinct components of a nucleotide? So what are the characteristics of a nucleotide? What are present in the nucleotide is what you have to write here exactly. So they are made up of what? One is they have a heterocyclic compound. The next is they have a monosaccharide. That monosaccharide can be a ribose sugar or it can be a deoxyribose sugar. And next is they have a phosphoric acid or a phosphate group attached to it, because at the end of the day nucleotide is made up of what? It contains a monosaccharide, that is a pentose sugar, or a pentose sugar, that is a ribose or a deoxyribose sugar, and it is that deoxyribose sugar or that monosaccharide or pentose sugar is attached to what? To a heterocyclic ring, and that heterocyclic ring is attached to what? To a phosphate group. Now these are the 3 components. So 3 components you mentioned there, 3 marks you will get. Next question, explain. So they have asked 3 questions here. So each will carry 1 mark. So you have to explain peptide bond, glycosidic bond, phosphodiester bond. So peptide bond, so where do you find them? Peptide bonds are usually found between 2 amino acids. So amino acids are linked with carboxyl group of one amino acid with the amino group of the other amino acid through a peptide bond. Next, glycosidic bond. Glycosidic bond, usually you find them in polysaccharides, wherein each monosaccharide unit is attached to each other through glycosidic bond. Next is phosphodiester bond. So phosphodiester bond, you find it between a phosphate group and then a sugar molecule, that is, and a monosaccharide. So monosaccharide is attached to a phosphate group through phosphodiester bond. 2 monosaccharides are attached to each other by glycosidic bond. Amino acids are linked to carboxyl group, its amino group and all that through peptide bond. So this is about the 3 different bonds. So you write about their linkages, you will get 1 mark each. So next question, what are essential and non-essential amino acids? Give an example for each. This is also an important question. Essential amino acids means they are needed in a diet. You have to take them as a supplement in the form of food and all that, because they are not produced in a body. That is essential amino acids. Example, valine, leucine, phenylalanine, etc. Non-essential amino acids means, so any one example you can give. I've given 3 here, which was there in the textbook. Any one you can write. So non-essential amino acids means which can be synthesized in a body itself. Therefore, you don't have to take it somewhere from other source. So it is called non-essential amino acid, like glycine. Then we have alanine, then glutamic acid, etc. So next question. What are apoenzymes? How they catalyze the biochemical reaction? Give an example. So 3 questions are there. If you write what are apoenzyme, you'll get 1 mark. Tell how or if you write how the biochemical reaction or how apoenzyme actually catalyzes the biochemical reaction, you'll get 1 mark. And if you give an example, you will get 1 mark. Totally 3 marks. So what are apoenzymes? Apoenzymes or apoprotein is an enzymatically inactive protein part of the enzyme. So in the enzyme, the protein part which is not active is called as an apoenzyme, and therefore to become active it requires a cofactor, that is an apoenzyme. How they catalyze, how apoenzyme catalyzes the biochemical reaction? So apoenzymes, first they are inactive. Then what happens? Cofactor will go and bind to there, making it active. So after binding to a cofactor, apoenzyme will become a holoenzyme, and this holoenzyme is nothing but an active enzyme which will catalyze the biochemical reaction. Example, trypsin, pepsin, urease. So any one if you give is enough. So this is about apoenzymes. So next question. Name the 3 kinds of cofactors. So the 3 kinds of cofactors are prosthetic groups, coenzymes, metal enzymes. So 3 you write, you will get 3 marks. Classify the amino acids based on, so classify the amino acids, read a question properly, based on number of amino group and carboxyl group, and you have to write 1 example. So based on number of amino and carboxyl groups, the amino acids are of 3 types: acidic amino acids, basic amino acid and neutral amino acid. So acidic amino acid example is glutamic acid. For basic amino acid example is lysine. For neutral amino acid it is valine. So you have to write one example for each of these. Next question. Give 3 examples of complex polysaccharides in nature. So you write 3 examples for complex polysaccharide, you will get 3 marks there. So one is starch, glycogen and cellulose. So 3 of examples you have to write for polysaccharides. So these are the important 3 marks question from this particular chapter. So this was about the session, and the revision class for this chapter comes to an end with this session. So I hope you found this useful, because it's quite useful for the exam. So meet you in the next session with a new chapter. We'll discuss the important questions under that particular chapter. So see you in the next session. Thank you.